All right. So we're going to start way back at the basic definitions. And hopefully it will be helpful to make a little bit more sense when we're talking about the same stuff. So remember, an antigen is any substance that specifically binds to a part of the immune system, an antibody or a T cell receptor. But not all antigens elicit an immune response, because if it's a self-antigen and it binds to a T cell or to an antibody, you don't want an immune response to happen, because that would be autoimmunity, and that would be bad. So not all antigens elicit an immune response. Antigens that do elicit an immune response are called immunogens, and I don't typically use that term, but that's the correct term that real immunologists use. So, so this is going to kind of be a flashback to the ACT, where they were kind of doing, plays with words. So an immunogen is always an antigen, but an antigen isn't always an immunogen. Okay, makes sense. So antigens are typically protein. Most of the time the immune system really likes to eat protein, but it can be a glycoprotein, it can be a glycolipid, nucleic acid, polysaccharide, and we'll see later on in this hour that B cells kind of like sugars, so sometimes those are immunogens as well. And it's actually kind of nice, because at the very end of this lecture, we're going to see that some bacteria think they're really smart, and they cover themselves with sugar, thinking, oh, well, the immune system likes protein, so I'm going to just coat myself with sugar, and then it'll be like my invisibility cloak, like Harry Potter. And but the immune system has figured out ways to recognize sugars, and they can be immunogenic, or lipids, and they can be immunogenic as well. So, but in general, the immune system really prefers proteins, and they, not just proteins, but they like proteins that are interesting. Like, you don't want a boring, monotone little, protein. They like stuff that's highly folded, stuff that has positive and negative charges, stuff that's interesting that they can really grab onto, and something greater than 1 kilodalton. Smaller than that are called haptens, and I think we had mentioned the word hapten, but we're going to talk about that in just a minute. So on bacteria and viruses and all the pathogens and tumor cells, there are a bunch of different surface proteins and compounds that can be antigenic. So one E. coli bacteria can have thousands of different antigens or epitopes on its surface that can be immunogenic. And that's kind of nice, because we're going to find out in a little bit that each B cell that makes antibodies will make an antibody specific to one specific epitope on the antigen. So one little bump on a pickle, and it's very specific to that exact little epitope or antigenic determinant. And since that E. coli has thousands of different antigenic determinants or epitopes on its surface, a bunch of different B cells can attack that pathogen, with different antibodies. And so it's a much more efficient way of getting rid of that bacteria, because there's all different kinds of bumps on the pickle that, different B cells can attack to be more efficient in killing that pathogens. Okay, so antibodies can be used in several different ways. They can be used to just neutralize the antigen, so they can just kind of grab onto their epitope that they recognize, and it's kind of like, okay, so I'm kind of in mommy mode. Sorry about that. So yeah, I'm kind of in grandma mode, mommy mode, baby daddy mode, so like, can you imagine, like, my grandson, grabbing onto his mom's ankle, and she's like, honey, I gotta go to work, and he's like, Mom, don't leave, I miss you, and she's trying to get to the door, and she's got this kid wrapped around her ankles, Well, antibodies can kind of do that. They can kind of neutralize a pathogen and just prevent it from being pathogenic. And the way they can do that is maybe preventing it from crossing the, a mucosal surface and gaining entry or something like that. Does that kind of make sense? So that's neutralizing an antigen. Some antigens can activate complement, and so remember we were talking about the complement cascade a while ago. There were 3 different ways that the complement system can get going, and one of them was the classical system where you have to have antibody production first, and then the antibody can bind onto some of those initial complement proteins and start the complement cascade. So here's where that's going to make a little bit more sense. Then they can also act as opsonins, the ketchup on the French fry, and kind of make the pathogen, antibodies can kind of bind onto the pathogen and make them a little bit easier to phagocytose and digest, so they can be opsonins, or they can promote antibody-dependent cell cytotoxicity, so they can be the vehicle for natural killer cells to grab onto the antibody-antigen complex and kill with ADCC. Okay. So I talked a minute ago about haptens. So what is a hapten? It's a small small antigenic determinant that can bind to an antibody, but it can't elicit an immune response because it's too small. It's less than 1 kilodalton. It's too small to elicit an immune response by itself. So they're antigens, but not immunogens. So that's 2 different ways that an antigen can not be an immunogen. One is if it's too small, the other is if it's self and the body recognizes it as self. So haptens are too small. So in order to make them become an immunogen, the hapten has to stick on to something a little bit bigger. So, it has to stick on to maybe a carrier protein like albumin. And then it's, it becomes more interesting because it's a little bit more 3-dimensional. It's something that the body can, relate to as an immunogen. So why is this important? Well, there's a couple different ways that this can be important. One that we'll learn about, coming up pretty soon is with poison ivy. So there's a compound in poison ivy sap called urushiol, and it is a hapten. So in and of itself, that little urushiol is not really very interesting to the immune system, but it binds onto proteins in the body, and so you get poison ivy sap on your skin, and some of the proteins on your skin bind that urushiol, and then that hapten becomes immunogenic and can cause a type 4 hypersensitivity response and elicit that itchiness and the blisters and the misery of poison ivy. Another example is a hapten. Some drugs like beta-lactam antibiotics are haptens. So in and of themselves, they're not immunogenic, but if they stick onto a carrier protein like albumin, once they're administered, then they can become immunogenic. And what happens if a drug becomes immunogenic? We can have an adverse drug reaction, or it can just inactivate the drug and then it's not helpful anymore. Does that make sense? Good so far? Okay. So this is just kind of an example of, the little hapten, and it can bind to the B cell, but doesn't really do anything on its own, so it's not immunogenic on its own. But if it is attached to a big carrier protein, then it becomes more immunogenic, and then the B cell can become a plasma cell and start making antibodies against that hapten. Okay. So like I said a minute ago, each B cell that becomes a plasma cell to make antibodies is programmed to make only one specific type of antibody against one very specific immunogen, one specific epitope on a pathogen. And like I said, each bacteria or virus or foreign cell or tumor cell has hundreds or thousands of possible epitopes on it or immunogens on it. So we have different B cells attacking different epitopes on that pathogen. So, and they all kind of work together. Each B cell produces a specific antibody against a specific epitope on the pathogen, and then it's friend B cell makes another form, another antibody against a different epitope, and so they all just, their armies all kind of come in from different sides and more efficiently attack that foreign invader. Does that make sense? Okay, so what the heck is an antibody? Let's talk about that a little bit, and we're going to talk about what an antibody is, how they're formed, and it's complicated, and then sort of what they're used for, and then I think we'll be done. Oh, then we're going to talk a little bit about how pathogens try to one-up the immune system, and how there's always this struggle between good and evil. We'll talk about that at the very end. So what is an antibody? So antibodies are either bound to the surface of an immune cell, or they're secreted proteins that kind of float around in the blood. They're produced by plasma cells, which are B cells that have transformed into antibody-producing cells. And they are kind of the key in the humoral adaptive immune system. So antibodies are proteins, and they're made up of 4 polypeptide chains. You have 2 identical heavy chains and 2 identical light chains. And so each Y-shaped antibody has 2 on the top, the 2 tops of the Y are the antigen binding areas that we call FAB, antigen binding, and those are called the variable parts of the antibody, and they're called variable because that's what varies to very specifically recognize that specific epitope on the pathogen. So you got 2 FAB regions that bind the antigens, and then the bottom part of the Y is called the FC, or constant region, and that's the part that binds to a phagocytic cell or other cells for like for ADCC. Okay, so we have different classes of antibodies. So and we'll go into this a little bit more here in a minute, but since antibodies have various different jobs, they might neutralize at a mucosal surface. They might be better for for phagocytosis. They might be just just act as a B cell receptor, or they might be produced either early in an infection or late in an infection. And so those different scenarios necessitate different forms of antibodies. So we have different classes of antibodies, 5 different classes. We have IgM, and this is IgM right here. It's called a pentamer because there are 5 antibodies all connected together with what we call J chains. So it's a pentamer, which means it has 10 different antigen binding sites on it. So this is an antibody that's formed really early on in a humoral immune response. So have you ever heard of, like if you're giving an antibiotic or a steroid or something, you often start with a loading dose, like you start with a real high dose, and then you kind of taper off? Have you experienced that yourself, or, in your shadowing? So this is kind of the same concept. We start out with this pentamer that has 10 antigen binding sites, so it's kind of the loading dose to kind of grab as many antigens as it can early on and kind of knock down that infection as quickly as possible. So IgM is one of the first forms of antibodies that are produced. So we have pentamers, we have monomers, and so the monomers are most commonly the secreted forms of antibodies. They're just one Y kind of out on its own, and it's kind of nice because it's small and it can go places. Some of the membrane-bound ones that act as B cell receptors are also monomers. And then IgA is kind of the unique one. It can be a dimer, which has 4 binding sites on it, 2 stuck together by a J chain, and they have 4 FAB regions, 2 and 2. And IgA is one, and we'll see here in a minute. IgA dimers are actually secreted onto the top of the mucosal surface, like in the GI system and the respiratory system. And secretory IgA, the dimer, is really good at neutralizing pathogens. So it binds the pathogens and prevents them from getting across the mucosal surface and causing infections. So it just kind of, keeps, it's kind of like I don't want to say border wall because that's a controversial subject, but that it's kind of the barrier to keep those pathogens from even getting in. Does that make sense? Yes. No, good question. So IgM, and we will get to this a little bit more in just a minute. IgM can be in a pentamer form. It can also be a monomer and act as a B cell receptor. So, good question. You took immunology, didn't you? Yeah. All right, good. I'm glad it's kind of coming together. Okay, so how does the immune system make these different classes of antibodies? It's the heavy chains that determine the isotype. So IgD is, see these are the five: IgD, IgM, IgG, IgA, and IgE. IgD, which is the B cell receptor, it's a monomer, and it has a delta heavy chain. So the heavy chain is delta, and that makes it IgD. IgM has a mu heavy chain, and that's either the pentamer or a monomer, which is another B cell receptor. IgG is kind of the most mature form of antibody, and it's a monomer, and it has a gamma heavy chain. IgA, that dimer that's on the mucosal surface, has an alpha heavy chain. And then IgE, the fifth type of antibody, it has an epsilon heavy chain, and it's involved in allergies or parasitic inflammation. Okay, so we talked about the FAB, talked about the FC region, we talked about the 5 different isotypes a little bit. We talked about the J chains that can make the dimers or the pentamers stick together. And then another feature of antibodies is it has a hinge region, and so the Y can be really skinny or it can be really wide because it has a hinge, like it has a joint in there. It has a rotator cuff in there that can go from either skinny or really wide. And the reason it does that is because it might want to grab onto two different pathogens that are a little bit farther away, or it might want to grab onto on the same pathogen 2 different epitopes on the same exact pathogen, and so it has some flexibility there so that it can grab as many epitopes as possible. Okay, let's see. So we talked about the monomers, IgD, IgE, IgG. The IgA, it can be a monomer in serum, but we don't really see much IgA in serum. Most commonly, IgA is a dimer that sits on the mucosal surface and acts to prevent pathogens from getting in. IgM is usually, most commonly, that pentamer. It can be a monomer and act as a B cell receptor on the surface of the B cell. And the multimers, the dimers or pentamers, are joined by J chains at the FC region. Is this making sense? Okay, good. All right, here's some more terminology, and hopefully this will be helpful. So we've talked about how each monomer of antibody has 2 different binding sites and FAB regions, and so a monomer has a valency of 2 because it can grab 2 different epitopes at the same time, so there's a valency of 2. So IgG, IgE, and IgD have a valency of 2. IgA, since it's a dimer, has a valency of 4, and IgM pentamers have a valency of 10. Does that make sense? 10 binding sites. So that's one factor in how helpful, how effective an antibody is how many antigens can it bind at once. Another feature of antibodies is how tightly can they bind the antigens. So some of the antibodies that are formed early on kind of loosely bind antigens, but don't bind them very tightly. And so antigens that are bound kind of loosely to the FAB portions have a lower affinity, so it's just like strength of binding. But as the antibody gets more and more mature, it binds tighter and tighter to the antigens. And by the time you get to that mature, the IgG or IgE or IgA, the mature forms of the antibody, then they have really high affinity and they fit just like a glove. And so that's high affinity. So, there's two different, things we have to come into, take into account when we're figuring out how efficient the antibody response is. One is the valency, and then the other is the affinity of binding. And so those taken together are called avidity. So avidity is kind of taking into account the affinity and the valency of the antibodies. So you can have, high affinity, but low valency like an IgG, or you can have low affinity but high valency with the IgM, and those all determine the avidity. Does that make sense? Okay. A lot of terminology. Okay. So this is just kind of a wrap up of the antibody isotypes and sort of their general functions in the immune system. So, we talked a little bit about IgA, mucosal immunity, and Dr. Johnson's going to talk about that tomorrow morning a little bit more. IgD is just the B cell antigen receptor. IgE is involved in parasite, allergic responses, hypersensitivity responses. IgG is sort of the workhorse of the immune system. It's one of the most mature forms of antibodies. And IgM is one of the earliest forms, so that one would have a lower affinity because it's kind of what I call a rough draft of the antibody. And okay. We will also see, and more as we get into some of the more clinical lectures, that antibodies have a really high affinity for their antigen. They're very specific. They like those three-dimensional antigens that have positive and negative charges. And by the time they're fully formed, like to the IgG form, they're very specific for one particular epitope, one particular antigen. Every once in a while, though, there is another compound, maybe a self molecule, maybe something else that's not pathogenic, that has almost exactly the same structure as that epitope that the antibody does recognize. And so it's possible that you can have cross-reactivity. So the antibody is targeting its particular epitope, but maybe there's another antigen that's a self-antigen or a non-pathogenic antigen that can fit that FAB region pretty well, and so you can have kind of an unintended response against that self-antigen or that non-pathogenic antigen, and that's called cross-reactivity. And we'll talk about that a little bit later in autoimmunity and things like that. So sometimes we also call it molecular mimicry. So that self-antigen or that non-pathogenic antigen is mimicking the pathogen that the antibody was made to respond to. Does that make sense? Yes? Yeah, if it's non-pathogenic and the immune system responds to it, then it doesn't really, it's not really clinically consequential. But if it's a self-antigen and it responds to the self-antigen, that's when it can become a problem, because then you've got a humoral immune response against self, and that's called autoimmunity. Does that make sense? Okay. Okay. So let's think a little bit more about the structure of antibodies. So we talked about them as proteins produced by mature B cells, which are called plasma cells, in response to an antigen that it can recognize. So again, antibodies have symmetry. They have two identical heavy chains, two identical light chains, and you can kind of see, this is a really good picture, kind of see the heavy chain is the inside of the Y, and then you've got those two light chains that are kind of on the outside. So the tricky thing, something that a lot of students think is, oh, the heavy chain is the FAB part, and the FCE part is the heavy chain, but in actuality, the light and heavy chain are both a part of the variable region, okay? And then the constant portion is down here. Does that kind of make sense? Okay. So, so I'm going to make you think a little bit more. So when you think about we talked about E. coli, and E. coli can have thousands of different epitopes on its surface. And so you could have 1,000 different B cells making 1,000 different types of antibodies against all of those epitopes that are on the surface of the E. coli. But when you think about, if you need 1,000 different types of antibodies just for E. coli, then you've got to think about, I need to be able to respond not just to E. coli, but staph and strep and pseudomonas and all the viruses and helminths and tumor cells. And so you need to have an infinite number of possibilities in the structure of antibodies to be able to fight off, anything that might come your way. Does that make sense? And so, what our body does, our immune system does, is it kind of custom makes each individual type of antibody to be exactly what we need. And so there are a bunch of different pieces that we can sort of glue together to make this antibody. And so we have these possibilities. It's kind of like going to an all-you-can-eat buffet, and you can have a little bit of this and a little bit of that and a little something else. And no, I really don't want to eat the seafood. It looks like it's been sitting out too long. And you kind of piece everything together and kind of glue it together, and it custom makes that antibody specific for that epitope that it's looking at. And so you get some of those alleles from dad and some of those alleles from mom, and actually you get multiple alleles from dad and multiple different alleles from mom. So you have you can piece them together in a very specific way to make each antibody custom made for the antigen that it needs to recognize. And so when we're putting the light chain together, the light chain has several different segments. It's called V, J, and C segments. And so you can pick one V and one J and one C from a whole smorgasbord of V's, J's, and C's to make that light chain. And then even more than that, there are two different types of light chains, kappa and lambda. And so you can already see there's a lot of creativity involved in making an antibody. And there's a lot of different choices that the B cell makes in order to pick, of all the V's, I want that V. And of all the J's, I just want that J, and so it picks, the specific parts that it wants to make its antibody. And it's the same thing with heavy chains. It has V, D, J, and C, so it has another part to it, the D region. And so that B cell, when it's still in the bone marrow, has this whole smorgasbord of V's and D's and J's and C's, and some from mom and some from dad, and it has to pick one of each to form its unique antibody. But once it picks, it can't change its mind. It can't go up to the buffet and say, oh, I want the prime rib, and then go back to the table and say, darn it, I changed my mind, I want chicken. It can't go back and get chicken. It's stuck with the prime rib, which is a good choice. Okay, so that, decision making is called allelic exclusion. So once it picks that one J, it can't go back and pick a different J. It's excluded all the other J's that were possibilities before. It's called allelic exclusion. Okay. Still good so far? Okay, so in order to make this custom-made antibody and make it just perfect and very specific for the epitope that it needs to bind to with very high affinity, and then to have the constant region be exactly what it needs for the specific type of immune response, there's a lot of steps that have to take place. And it's very unique so this type of protein, this antibody protein, is made differently than most proteins. Most proteins just go through the transcription, translation, post-transcriptional modifications, and then it's done. Antibodies need to go through a lot more steps in order to really be exactly perfect. And so I always think about antibody formation as sort of like, remember when you were in college and in English class, and you had to write a term paper? So you kind of write an outline, and that's allelic exclusion, and then you kind of write a rough draft, And then, you sleep on it and you look back at the rough draft again and go, oh my gosh, that's terrible. And then you have to keep fine tuning it, you make grammatical errors and, make sure there's subject verb agreement and make sure that, you have the right number of sentences in your paragraph and everything. And so it takes it takes several rounds of kind of, editing that term paper before it's ready to turn in and you get the A, right? So that's kind of how I think about antibodies being formed. So there's a few steps that happen in the bone marrow before that B cell even gets released from the bone marrow. And then there's some very important steps that happen in the secondary lymphoid tissue after exposure to the foreign antigen. So these are the steps we're going to talk about here in a minute. We're going to talk about selection of the heavy chain, and then we're going to talk about selection of the light chain, the kappa or lambda light chain. So we have the kind of that structure, the skeleton there, the heavy chain and the light chain. And then it goes through the allelic exclusion. So picking those B, D, J, and C alleles from the smorgasbord, allelic exclusion. And then it just kind of, then by that time it's kind of tired, and so it just kind of throws things together, and sometimes it's kind of sloppy. And so then it goes through a basic receptor editing where it kind of splices out things and kind of rearranges things a little bit, and that's with RAG. And so then it kind of has a rough draft. And so that rough draft is going to be either IgD or the IgM monomer, and it's going to be bound to the surface of that naive B cell. And so that's kind of the initial rough draft, the sloppy puppy, okay? And once it goes through all of that, then the B cell is considered mature but naive. So, this is kind of its adolescence. So this is your daughter, and it's going through all of its, all of her, growing up. And when she gets to this point, she's and you'll understand when you get to be a parent. So she's maturing, and she's, kind of determining who she's going to be when she grows up. But man, you are not going to let her date until she gets to be 16, right? And so she, by the time she gets through the RAG step, she's mature, but she's still naive. She hasn't had her first kiss yet. And so then you're, okay, I've done the best I could, I've raised you as much as I can, and now it's time to go out in the world. So your little girl, your little 16-year-old girl who's mature but naive, goes out into the secondary lymphoid tissue, and there she meets those boys. She meets those bad boys, those antigens. And and there, once she meets her, those bad boys that you don't want your daughter to date, then she has to make some adjustments. And so she's gonna keep working and kind of modifying until she's just perfect, and she finds that, just that right fit, and she falls in love and gets married. So in the bone marrow, they get to the point where the B cell is mature but naive because it hasn't been exposed to pathogens yet. And it gets to the point of making the IgM or IgD as a B cell receptor, and then it goes out into the secondary lymphoid tissue, and that's where it can be exposed to pathogens. Is that good so far? All right. Okay, so you know this by heart now. So you have pathogens that are recognized and phagocytosed by antigen-presenting cells. They digest it. Remember we talked, the very first day when you thought immunology might be cool. The phagosome fuses with the lysosome, digests it in the phagolysosome. And then remember I said, and they kind of keep the interesting parts. The interesting parts are the epitopes. And so it keeps the interesting parts. It presents the antigens to the adaptive immune system, and then the Th2 cells stimulate the B cells to kind of start going into plasma cells to make antibodies against that antigen. So that all happens in the secondary lymphoid tissue, in the lymph nodes, in the spleen, in the Peyer's patches, in the appendix, in all those secondary lymphoid, in the bulk, gulp, all of those, to make antibodies against that antigen. And then, the mature forms usually get secreted and they go out into the blood and they look for those exact epitopes that they've been trained to recognize and bind them and, target them for complement destruction, opsonization, neutralization, all that stuff, ADCC. Okay, so this is kind of what we just talked about, is what happens in the bone marrow. So you have the pluripotential stem cells that can become anything. Then they start doing start picking D and J. You don't really need to remember all these specifics, but just to kind of hopefully get things in your, clear in your head. Starts picking the D's and J's, and then it starts picking the V's in addition to that. And then it makes that pre-B cell receptor, so that IgM or the IgD, which is the rough, sloppy copy. And see, at this point, it has a mu chain, so this would be IgM if it has a mu chain. And then it goes to, the BD and J are rearranged, and on the heavy chains, then the light chains start working, and they get their V's and J's rearranging. Then when those are rearranged, then IgM is expressed on the surface, and IgD is expressed on the surface, and so those are the two rough drafts, possible rough drafts in their B cell receptors. At this point, your little 16-year-old girl is mature but naive and ready to go out into the secondary lymphoid organs. Okay, good. And I think this just kind of shows the same thing, but in a little bit different way. Yeah, that's pretty much the same thing. Okay, so the mature naive B cell has receptors on its surface, and it's either IgM or IgD. Let me remember it now. Okay, so now we're going to activate those mature B cells. So the mature naive B cells go down to a secondary lymphoid tissue, and then they just kind of sit around and wait for an antigen that they can kind of at least bind with low affinity. So they bind with low affinity, and then the action starts happening. Okay. So in order for a really effective B cell immune response, you have to have at least that low affinity binding with the pathogen. You have to have cytokines from those helper T cells being produced, and then you also have the co-stimulatory molecules that Dr. Johnson's talked to you about to really have a vigorous, effective humoral immune response. Okay. So as that second half of development occurs in the secondary lymphoid tissue after antigen exposure, those B cells that have met their bad guy boyfriends for the first time, they have to go through a lot of transformation in order to become a mature plasma cell and produce those mature antibodies. And so what's happening is they have to, they get exposed to the antigen, they kind of loosely bind it. Then what they're going to do is they're going to start mutating that FAB region. So, and it's called, we'll get to this in just a minute, it's called somatic hypermutation. So you know what a somatic cell is. We've got somatic cells, which are, all the cells in the body. And then you have germline cells, like eggs and sperm. And so somatic hypermutation is a really rapid succession of mutations of this B cell that changes the affinity of the FAB portion of the antibody that it's producing. Now, in general, we don't want cells to hypermutate. If you have a skin cell or a liver cell or a pancreas cell that's hypermutating, that usually means cancer. So usually hypermutation is bad, but the B cells are very unique. They're designed to hypermutate, And so they go through rapid mutations of the FAB region, and then they divide, and they have two daughter cells that are producing a slightly different form of the FAB region, and then they mutate and divide again, and mutate and divide again, and so what's happening is you're getting more and more cells in that follicle, in that secondary lymphoid area, and so as you have a developing humoral immune response, that follicle where the B cells reside is going to get a little bit bigger, a little bit more active. You're going to have a lot more mitotic figures in there, and that's when that follicle becomes a germinal center. Remember that from when we were talking about the organs of the immune system? So an activated follicle where B cells are trying to respond to an infection by mutating and dividing and increasing the affinity of the FAB regions, that's when you get a germinal center. So here's I thought I had a picture of the germinal center. Maybe it's coming. So, okay, so just to kind of recap a little bit, we have a lot of different ways that we can introduce antibody diversity. And the first one in the bone marrow is that allelic exclusion, B, D, and J. You pick the 1B, 1D, and 1J out of a whole bunch of possibilities, and C as well. And any of the ones that were not chosen by that B cell in the bone marrow are excluded, and that's called allelic exclusion. Okay. Then we also have, and this is before we leave the bone marrow, we have rearrangement of the antigen receptor genes in the B lymphocytes. So this is where RAG comes in and kind of splices and kind of, reorganizes and fixes the sloppy copy. And that kind of helps to generate a little bit more diversity, and also makes it a more efficient antibody. Okay, and this is where recombination activating genes, or RAGs, are used to kind of splice out the junk and make it a little bit more efficient. Okay, and so as we get going, we want to make sure that the developing B cell is not responding to self. And so we had talked about positive and negative selection in the thymus for T cells. We have receptor editing with B cells that kind of helps to eliminate those self-reactive B cells. Okay, so and then when the B cell has reached maturity, it has a B cell receptor, IgM or IgD, and that's a B cell receptor that's membrane bound on the B cell surface. And that all happens, the selection of heavy chain, selection of light chain, and the allelic exclusion and the receptor editing. All of that happens before she leaves the bone marrow. And so she's a mature, naive B cell when she leaves the bone marrow. And then she goes to the secondary lymphoid tissue and meets her pathogens that she has low affinity for. And that's when these two steps happen. So we have somatic hypermutation and we have isotype switching. And so, we talked a little bit about somatic hypermutation. It happens after exposure to antigen. So you have sort of a low affinity binding of that immature antibody, that low affinity antibody, the IgM or IgD. And so it kind of binds with low affinity, and then that B cell hypermutates the FAB portion, divides into two daughter cells, and that mutation may have resulted in an FAB region that has higher affinity for the antigen, or it could have made a mutation that makes it lower affinity. So if the daughter cells of the B cell make a mutation that is worse than before, then that daughter cell dies by apoptosis. But the ones that undergo somatic hypermutation and have a better FAB portion, higher affinity for the antigen, then they get to survive and then go through hypermutation again and again and again. So over the course of a few days, that mature B cell is keeps going through more and more rounds of hypermutation, and only the improved FAB regions get to survive. The ones who are producing FAB regions that are lower affinity have to die by apoptosis. And so you go all the way through somatic hypermutation until you get a perfect fit, and then somatic hypermutation is done. And so then the FAB portions are perfect. And then the last step is to kind of figure out which FC portion we need. So, if the FAB portions are working on an allergen or on a parasite or helminth or something like that, then we're going to need an epsilon heavy chain for the isotype that we need. And so that final step is isotype switching, where we pick which heavy chain would be most helpful for the specific humoral immune response we want. If it's allergy or parasitic infection, we want epsilon, and that isotype is made for IgE. If we just want a general workhorse, then we're gonna isotype switch to a gamma heavy chain, and then you make IgG. If you want mucosal immunity, then you get an alpha heavy chain, and isotype switch to alpha chain, and then you make IgA dimers that go on the cell surface. Is that good?
Right, for IgA, since it goes to the surface, but it's made in response to RNA countering the pathogen,
Mm-hmm.
does it just stop more pathogen from coming in, or can it also?
Yeah, so IgA can be a monomer or it can be a dimer. So in the secondary lymphoid tissue, there's probably enough of the infection that has gotten into the secondary lymphoid tissue, been presented. So back up a minute, slow down, operator, speak English. So, the antigen presenting cells that are like in the gut, for example, they will, grab, because they're always surveying the mucosal, surface. They will grab some of that pathogen, process it, take it down to the regional lymph nodes, like the mesenteric lymph nodes, for example, and they'll get the B cells to become plasma cells and make antibody against that pathogen that they found at the mucosal surface. Then most of the IgA is going to be pumped back up to the mucosal surface for neutralization of pathogens at the mucosal surface. Some can circulate around in the bloodstream because if you may already have entry of some of those pathogens into the circulation. So there's a little bit of IgA that circulates around in the system. Most of it's going to be pumped back up onto the cell surface. Right, and the serum is the monomer on the mucosal surface is a dimer. Does that make sense? Okay, good questions. I hope I'm making sense. I hope it's kind of all fitting in a little bit. And we need to take a break here in a minute. Do you want to finish our thought right here, and then we can take a break? Okay. So I think we've already gone over this pretty well. So somatic hypermutation occurs after antigen exposure in the secondary lymphoid tissue, and each generation of B cell has point mutations that may be better affinity or lower affinity for the antigen. And it's a very high rate of mutation, which is very unique in the somatic cell lines. And so this process of getting, each successive generation of B cell, hopefully getting better, a higher and higher and higher affinity, that's called affinity maturation. And once somatic hypermutation and affinity maturation are complete, then we have isotype class switching to make the specific type of antibody that we need. All right, do you want to take a quick break and then we can finish up real quick? Okay, let's take a little break. If you have questions or if I did